Okay. So our group is presenting on genetic engineering, specifically cloning. We're Team Fidelity, and presenting is going to be New, Leslie, Regina, and Hunter, and myself. So just to start on the topic, genetic engineering is defined as the altering of genetic makeup of an organism. There are many different forms of genetic engineering. There is genetically modified organisms, also known as GMO, where their genome has been altered and artificially changed. Some examples of GMO organisms are fruits, vegetables, animals, and meats. There's also cloning, which is what our main topic is about, which is creating exact genetic copies of genes, cell, or an entire organism. So this is where the genetic makeup, that is DNA, is identical. So researchers today have the capability to clone genes, cells, tissues, or entire organisms such as sheep, dogs, or monkeys. And this has been done before. And then there's the natural form that occurs through nature, which is like identical twins. And this is the exact same DNA. So currently in the United States, uh, cloning is not legal in 15 states where it's banned completely. However, there is no federal law that prohibits human cloning, but the few of these states that prohibit it completely is states like California, Florida, and Georgia. But yeah, the Patient's First Act of 2017 allows for stem cell research from cells that are ethically obtained.
So for the procedure, um, in human, firstly, the scientists will make a clone by transferring the DNA from the animal is somatic and play into the egg cell with nucleus and DNA removed. The egg develop and become an embryo that contain the same gene as the cell donor. Then the embryo is implanted into the woman uterus. So here is the 5 step of cloning in the plant. So first, DNA extraction, the extracting DNA from the organism. Step 2, gene cloning. So we select the gene, a gene for cloning. Step 3, gene design. So we replace an existing promoter with a new promoter. 4, transformation, using the gene gun method to transform plant cells. 5, black card breeding. So right here is the last picture. And I also provide here the human cloning. So basically human egg donor, giving the donor egg and remove nucleus that I mentioned earlier. So there's a fuse egg and cell with electricity. The scientists gonna do transformation, and so we have an egg fused with cell and implanting embryo into the mother. I'm done.
All right, so there can be many complications associated with cloning. It can be very inefficient. So Dolly is one of the first animals that was cloned. Dolly was one out of 277 embryos, so only one out of that. It's a very low percentage for cloning to actually happen. And not only was it a very low percentage, Dolly also had a lot of major issues. So she had deformities in some of her vital organs, such as her liver, heart, and brain. And some of the research shows that stem cells versus cancer cells, there's not much research pointing to the difference in the two as far as the beginning stages, because they both can replicate. So some of the clones have a very high rate of forming cancer. And also testing on human embryos leads to destruction in most cases. So it's so inefficient, and using so many different human embryos, it can almost seem like a waste because you're using so many and only maybe one or two will take from it. So there's many benefits and risks associated with cloning. And in some cases, the animal comes out okay and doesn't have many side effects. But in Dolly's case, there is many side effects, such as the complications with the vital organs. So it's important to take into account that the risks outweigh the benefits in the case of cloning.
So ethical issues associated with the cloning. So some of the um, ethical issues specific to human cloning include the safety and the efficacy of the procedure. Cloning, cloning for destructive embryonic stem cell research that the effect of reproductive cloning on the child or parent relationship and the commodi- com- commodification of human life as a research product. Another ethical issue is with the cloning is could be systematical elimination of sex or gender. In some countries like China, whereby whereby boys are more desired than girls, cloning could lead to pro- this country producing males at any alarm rates with no regards to female offspring. Another ethical issue could be with productive cloning lies not in its sexual characters, but it is also to on the understanding of on children as a gift rather than oppos, oppressions. Sorry.
All right, so for faith integration, we have verses from Genesis chapter 1, verses 26 to 27. Then God said, "Let us make mankind in our image, in our likeness, so that they may rule over the fish in the sea and the birds in the sky, over the livestock and all the wild animals, and over all the creatures that move along the ground." So God created mankind in his own image. In the image of God, he created them. Male and female, he created them. So while the Bible does not specify any subject related to human cloning, there are principles in scripture which may shed light on that idea. Cloning requires both DNA and embryo cells. If the cell accepts the new DNA, a duplicate embryo is formed. However, the embryo cell may reject the new DNA and die. While it is possible for a duplicate creature to be created in this manner, like for instance Dolly the sheep, the chances of successfully duplicating a creature without variations and without complications are extremely low. The Christian view of the process of human cloning can be stated in some verses in the Bible. Human beings are created in the image of God. Therefore, humans are unique, just as what Genesis chapter 1, verses 26 to 27 declared, that man is created in God's image and likeness and is unique among all his creations. Human life is something to be valued and should not be treated like a commodity to be bought and sold. Several people have advocated for human cloning for the purpose of creating replacement organs for people in need of transplants who cannot find a suitable donor. But the process of cloning requires human embryos to be used. While cells can be generated to make new organs, it is necessary to kill several embryos to obtain the required DNA. Essentially, The cloning would discard many human embryos as waste material, eradicating the chance for those embryos to grow into full maturity. So this is from Got Questions website. Thank you.
What do I expect?